right, these are going to be short answer notes part 2 for your study guide. So, what is the function of phospholipids in the membrane? The phospholipids have a hydrophilic head and a hydrophobic tail. So they are responsible for controlling what enters and leaves the cell based on their structure. So things that are small and non-charged can get through, but things that are large or charged, like water or ions, cannot get through. Two functions of proteins in the membrane. One of the functions of proteins can be for recognition. They can recognize other cells, or they could be used for other cells to recognize them, or for binding. They can also act in active transport to pump things through from a low concentration to a high concentration. They can act as channels in passive transport to allow things like water or ions to move from high concentration to low concentration. Those are always open. Those are just a couple of functions that should be on your study guide. Another one that I didn't go through. What is meant when cell membranes are described as being selectively permeable? I kind of discussed this in the first question, but it's just the fact that because of the structure of the phospholipid bilayer, the membrane only lets certain things in and keeps other things out. So it regulates what enters and leaves the cell. Next question, what does the term structure equal function? How does that relate to an enzyme? Every molecule that we talk about is a three-dimensional molecule, and so the structure of it is its areas that are polar, or its areas that are non-polar, or different bonding sites that are available based on its molecular arrangement, based on the atomic arrangement in the molecule. So, and then how each part of the molecule is joined together. So in an enzyme, you've got your primary structure, which are your beads of amino acids. You've got your secondary structure, which is how those beads of amino acids will kind of like bond with themselves, not the R groups, just hydrogen bonds. So they'll either make the spiral or they'll make the zigzag. And then you've got your tertiary structure, which is how those R groups start to interact and cause the folding. So you will have regions that are polar, you'll have regions that are non-polar, and you'll have a different shape depending on what amino acids are present. And then your quaternary structure will be how those different strands of amino acids, the different tertiary structures, will actually come together and make a more complicated, larger structure. So a lot of enzymes are functional at the quaternary structure. Many of them are tertiary. But the idea is structure to function. So if something happens and it causes a different structural point, so it denatures your protein in some way, it will maybe add a hydrogen and cause a change in shape, or heat comes and it breaks it down and it denatures it, or pH would be the hydrogen example. But whatever's going on, if it causes that structure to change, then the function no longer works. So the enzyme only works properly if its bonding site with the substrate remains the same, remains active in the way that it should be, and if anything happens that causes that bonding site to change, its structure no longer works, so it can no longer do its function. Next question. It says, why would treating cancer with Taxol, which prevents microtubules forming, be effective? So microtubules are what causes the spindle fibers to form. They're part of the structure of spindle fibers. And if you can remember from mitosis, spindle fibers are what attaches to the chromosomes, the sister chromatids, and they will attach at the centromere point and they'll pull those apart, and as they wind up, they pull them apart. So in order to prevent cancer, if you can cause the cells to stop being able to do mitosis, then they can't split apart anymore and they can't divide. So you cease cell division, and so you cease the growing of the tumor or the metastasis of the cancer. And what kind of gene is p53? I had this pulled up. You have a bunch of p53s, or types of p53s in your cell. They are basically tumor suppressor genes, and it says, if you just Google it, the simple definition is a gene that makes a protein called tumor protein p53. This protein is found inside the nucleus of cells, and it plays a key role in controlling cell division and cell death. It helps keep abnormal cells, including cancer cells, from growing. So kind of like as a checkpoint for cells that allows them to see if their growth is going to be abnormal and cease that if it starts having issues. But I do encourage you to Google that and look into it more. There's all kinds of different pictures and stuff about what a p53 looks like and how it works. But your basic idea is just that that's the gene that kind of is the checkpoint for, are we doing okay, or do we have abnormal growth, and what do we need to do to stop that abnormal growth. So I'll give you another video, and it will cover the, oh, I skipped one, I'm so sorry. You can't like wave at me or anything on YouTube. So we have one, number 9 on your study guide. It says, the cell in the picture was in a hypotonic solution. Label the following. The concentration of the solution, it's either 400 or 100. And the concentration of the cell is 400 or 100. And I don't really know, because I don't think that's supposed to be an O. So, but I think it's molarity of osmotic pressure. But anyway, it's either 400 or 100 in the solution, or 400 or 100 in the cell. And then you need to tell the direction of water movement and what will happen to the cell. Will it swell, or will it shrink, or will it burst? Swell means it'll burst, shrink, or it'll stay the same. So if it's hypotonic, I'm not going to give you the answer, I'm going to make you think about it, but if it's hypotonic, that means there's more free water outside and less stuff outside. And then inside the cell, there would be less free water and less stuff. So the water movement will always go from where there is less free water to where there is more free water until equilibrium is reached. And so hypotonic solution generally cause water to rush into the cell. It'll cause the cell to swell and burst. If you are an animal cell, if you're a plant cell, it'll just fill up and make the cell really full in that central vacuole. Well, plant cells don't burst because of their cell wall. Eventually, osmotic pressure regulates and it can't bring any more in. If a hypertonic solution, it'll cause the cell to wither and shrink. So if you have too much salt in your blood, that's bad for you because your red blood cells will start to shrink and shrivel up. If you have too much water in your bloodstream, it can cause your red blood cells to burst. So in an isotonic solution, which is one where both the concentration outside the cell and the concentration inside the cell is equal, then your cell will just stay the same and be happy, which is why you have 0.09, I believe, I could be wrong, but I think it's 0.09% saline solution inside an IV bag, so that the amount of salt in the IV matches the amount of salt in your bloodstream. So that'll be it for your notes for this section, and then we will cover multiple choice in the next one.